Today's webinar is organized by Environment Climate Change Canada, along with the Mackenzie River Basin Board, who we have Paula Civic here, who is the Secretariat of the Mackenzie River Basin Board, and will be giving us a quick intro in a second. Today's webinar is going to be focused on research and community-based monitoring that's ongoing in the Great Slave Sub-basin. So if you look at the State of the Aquatic Ecosystem report that I put in the chat, and that we'll link to later in emails and stuff and correspondence, the Great Slave Basin is one of many basins, as many of you know, within the Mackenzie River Basin. And as Paul will dive into a little bit, I'm sure, it is a vast and large basin, so it's very exciting to be kind of tackling it one sub-basin at a time. I'm here joining from Edmonton, which is on Treaty 6 territory, actually a little bit south of the basin itself. And for folks who are calling in today, if they want to write into the chat, hopefully this chat function totally works. I haven't done, oh, awesome, it does work. I see people talking. So if you want to put into the chat where you're calling in from today or where you're joining from and the Indigenous territories or the treaty lands that you're on, that would be really awesome. And we could see some, I guess, the breadth of where people are calling in from today. We also just, with our work here, want to acknowledge and honor the fact that Indigenous peoples and communities have been stewarding this basin for time immemorial, and from the headwaters in the mountains through the boreal forest and the tundra and to the outlet at the Arctic Ocean. The Mackenzie Basin continues to be and has been stewarded by many different Indigenous people, and we hope that through our work today, we can continue to kind of build on and support some of that work, stewarding the basin for ourselves and for future generations. So yeah, hopefully this webinar can be a small step in building towards some reconciliation and reflection. We have a really awesome list of presentations lined up today. They're kind of all about around fostering. Okay, Robert, I do not hear any audio. I think I'm getting some answers from other people, so I do think, Robert, it might be a problem on your end, but I think I would check down at the bottom. There seems to be some settings around audio output and input. But yeah, so we have a bunch of awesome presentations set up today, all focused on kind of collaboration and bridging knowledge gaps in the Mackenzie Basin, as our webinars have been kind of prompted by, and that all is kind of associated with the State of the Aquatic Ecosystem report that the Mackenzie River Basin Board puts out. So Paula will talk to that as well a little bit. But just to briefly go over our agenda for today, we're going to have some words from Paula. Then we're going to be followed by three different main presentations who are going to offer different perspectives on the basin. We have Kathleen Fordy from the Deninu First Nations Aquatics Department, and apologize if I said that partially wrong. I was practicing a little bit. But and then we have Jeffrey Cederwall from the GNWT, and we have Dr. Marlene Evans as well from Environment Climate Change Canada, as well as many other big pieces of work. Yeah, and following those presentations, we're going to have a short question and answer period. And the way that's going to work is if folks look along the bottom of your screen, there should be a Q&A kind of box that you can enter in questions. And folks, the presenters along the way can answer some of those questions via text if they'd like, the panelists that are here, but I'll be pulling some of the ones that kind of encapsulate some of the broader questions and sentiments, and we'll chat about those as a panel at the end. So going from there, I'm going to pass the mic off to Paula for some words on the Mackenzie River Basin.
Good morning, everyone. Hi, my name is Paula. I am in Edmonton at the moment on Treaty 6 territory, and I'm really, really pleased to see such a great attendance. Looks like there's 62 participants on this call at the moment, and I'm so pleased to see that. So as Carter set up, the Mackenzie River Basin Board has a duty to report on the state of the aquatic ecosystem of the Mackenzie River Basin. And the MRBB published an online report in 2021, and I will screen share for a second just to show those of you who haven't seen the report what it looks like. Can everybody see the screen? Yeah, great. So this online report shares information from different knowledge systems on 4 aquatic indicators in the Mackenzie River Basin. They are water quality, water quantity, health and well-being, and habitat and species. And right here I've got the page up, the summary page up for the Great Slave Sub-basin, which is the basin that we're focusing on today, or part of the basin that we're focusing on today. We're proud of this report, but there's lots of gaps and there's lots of information that we want to make sure that we capture or include in the next version of the report. We're always looking for ideas and information to make this a better, stronger product. And especially like when we talk about this part of the basin, the Great Slave Lake has seen some historic highs and historic lows just in the very small window since this report, since we finished the data gathering part of this report. Like it's incredible the amount of variability that we've seen in water in this part of the basin and in the rest of the basin as well. If you haven't had a chance to go to the report, I invite you to do so. You'll see a summary of the information that we are sharing for each of the indicators in each of the 6 sub-basins. There's an overview for the whole basin, for those that are interested in that. And in each, once you delve into the specific indicators a little bit closer, you get a sense of the detailed information that we've shared and some of the data gaps that, that we still need to fill. So these webinars go a long way to both filling some of the data gaps that we've identified and that we're looking to, support and enhance in the next version of the SOAR, but also to connect people who are interested in water in this massive geography in Canada. So I'm really thrilled we're here. I'm going to try to stop sharing, but I will say, Carter, that you got really small once I shared. So if you could stop sharing on my behalf, that'd be great. Oh, here we go. Stop. That's better. And thanks. I'm looking forward to the presentations today.
Awesome. Yeah, thank you, Paula. Just a reminder again before we get going, if you want to throw any questions into the Q&A, that's where we'll kind of be stocking them up. If you want to just put in brackets at the end, if you really don't want to be put on video, I can invite folks to be on video to ask their question when we get to that question period at the end. So just maybe make note of that at the end of the question. So we're going to get started with our first one today, and that is with Kathleen Fordy. And I'll let you do a bit of introduction for yourself, but I had to pass the mic off to you.
Hi, good morning, everyone. My name is Kathleen Fordy. I am calling in from Fort Resolution, Treaty 8. I work for the Deninu First Nation here as the Aquatics Coordinator. So I will share my screen and my Can everybody see it? Good. Okay. All right. So the Deninu First Nation. So this is the Resource Management Department, Aquatics Division, Aquatics Coordinator, which is myself, Kathleen Fordy. So grassroots and partnerships, the DKFN partners with Acacho Territory Government, the ATG, Acacho Aquatics Monitoring Program, AMP, Acacho ISETs, Fort Resolution Métis Government, Hamlet of Fort Resolution, the Government of the Northwest Territories, GNWT Water Resource Division, Dr. Genois, Department of Fisheries and Oceans, Dr. Marlene Evans, Environment and Climate Change Canada, Dr. Haley Hung, Environment and Climate Change Canada, Dr. Brenda Parlee, University of Alberta. So for our monitoring program, community concerns have always been Is the fish healthy to eat? Is the water safe to drink? Is the fisheries sustainable for generations? So we have the Little Buffalo River Angler Surveyor. So the DKFN LBRS monitor hands out and collects sport fishermen surveys from May to September. The survey records are scanned into PDF files and entered into Excel spreadsheets. The information includes where the fishermen live, what species they caught, and how many. Fish monitoring. So we collect dorsal backfin from pickerel for aging. The data that's, as you could see on the picture at the bottom. It's fish weight and length, and we enter that into Excel spreadsheets. And then the dorsal backfin, we send them off to DFO in Hay River. So the water part of the LBR surveyor. So the Little Buffalo River weekly water quality monitoring, which I attend, we call it our field day. So every Friday I go out and help with this as well. We go out and do with the YSI basic features such as temperature and conductivity, water samples for additional analysts. So what we do is each Friday, we do a routine, and every fourth sample, it's a routine. Nutrients and total metals we do as grab samples. Ramona Fordy has been doing this work for DKFN since it began in 2004. She is reliable with great traditional knowledge of the land and water. Ramona is the second generation monitor to assist in water and fish monitoring. Resolution Bay and Slave River. So the DKFN Little Buffalo River Surveyor has participated season routine trips with the government of the NWT Water Resource Divisions, community-based water monitoring program. So samples, we call it CBM, community-based monitoring. The samples are taken from 5 separate locations identified by local community members. So there's the Big Eddy on the Slave River, the mouth of the Slave River, and what we call camp. It's quite a ways up the Slave River, maybe about halfway to Smith from here. It's just above where majority of our community members have cabins and stuff. And then we have the Resolution Bay area, and then the mouth of the Little Buffalo River. So for the DKFN Aquatics Coordinator, my responsibilities as the Aquatics Coordinator is a variety of tasks. DKFN has trained Elizabeth Drew, Nicole Mackay, and myself, a third generation monitor members from DKFN members from Fort Resolution to conduct basic water sampling, entering of data, also surveys, trip reports, incident reports, and error check, analyze and graph, coordinate proposals and reports, training programs for the community. So I also do some sampling at the water treatment plant. So in order to address questions about the drinking water supply from Resolution Bay, I have been conducting studies of the untreated water that comes into the plant. Mind you, this hasn't taken place in a couple of years. This is the other part. So we copy the water treatment plant's intake raw data and enter it into the DKFN database. This allows for investigations of changing conditions in the Fort Resolution Bay for generations to come. Collect water samples for chlorophyll analysts weekly. Samples are analyzed by Dr. Marlene Evans' lab in Saskatoon. In summer, the water has more turbidity and takes longer to process through the filter. Conducted a special study of water released from the treatment plant into the Resolution Bay. As you see in the bottom corner, that's where we were sampling. we did switch that sample site as we weren't finding anything. So Dr. Marlene Evans, Environment and Climate Change Canada, has been a critical instructor for this position at the First Nation. Sorry, my phone started ringing. What's going on? Sorry, they're not supposed to be transferring calls to me right now, and they are. So the Resolution Bay, so DKFN Chief and Council have selected 2 locations, the Fort Resolution Bay area of the Great Slave Lake and the mouth of the Little Buffalo River, where routine collections of water samples are taken. Sorry, I kind of lost myself there. So the Resolution Bay area on the Great Slave Lake and the mouth of the Little Buffalo River, where routine collections of water samples are taken for monitoring trends of nutrients and total metals. The trip records and incident reports are archived. At the Little Buffalo River, we also collect data of water quality, weather, trip, and incident reporting. All the raw data are scanned and then entered into an Excel spreadsheet in the DKFN database. We now have the location at the Back Bay, which is across from our local dump / lagoon. We no longer take the samples at the Resolution Bay area anymore. Oh, what's going on? Oh, there we go. So we also do air monitoring for mercury and other chemicals. So I'm also responsible for collecting passive air samples at the location of Mission Island, as well as the new site just past Little Buffalo River. This is done every 3 months for the plastic jar sampler. It used to be like a puff disc sample, as you see the top right corner. And once a year for the XAD resin sampler. So samples are sent to Toronto. A poster for the initial results has been sent to the community. See, if you can see on top. This work is done in collaboration with Dr. Haley Hung, Environment and Climate Change Canada, and Northern Contaminants Program. Integrated eco-monitoring and assessment of cumulative impacts on Great Slave Lake fisheries ecosystems. So the DKFN Aquatics Division also works with Dr. Genois, Department of Fisheries and Oceans, on a multi-year research project. So a lot of it took place from 2012 to 2017, but we also were working with them past 2017 up to 2021. Provided coordination for logistical support, recruiting skilled workers to assist in daily operations, net setting and pulling, fish sampling, camp maintenance, and boat transportation. AFS and AROM funding is utilized for DKFN's participation. These studies will help better manage the commercial and domestic fisheries. Environment and Climate Change Canada and Northern Contaminants Program. DKFN has supported Dr. Marlene Evans' research project titled Spatial and Long-Term Trends in Persistent Organic Contaminants and Metals in Lake Trout and Burbot from the Northwest Territories. Each year, DKFN hires a member, Gabe Lafferty, first-generation monitor, to go out on the lake and set a net, collect burbot and pike, and submit those fish to Marlene's lab in Saskatoon, Saskatchewan for processing. Note that Gabe has been collecting fish for this project for over 20 years. Genetic assessment of Inconnu in the Great Slave Lake. So this work was done before I even started as the aquatics coordinator, which was in 2015. This was before I was here. So Inconnu are highly valued fish by community members and sports fishermen. In collaboration with Dr. Yaman and Ross Tallman, Fisheries and Oceans Canada, DKFN has participated in traditional knowledge surveys specific for Inconnu, assisted in field sample collection of in canoe on the Tulsin River. Tracking changes. So this was also done before I started in 2015 as well, in collaboration with Dr. Brenda Parlee, University of Alberta, and Acacho Aquatics Monitoring Program to identify key indicators with elders and youth to track the changes in the environment. Slave River Transboundary Fish Monitoring. So Transboundary Water Agreement was signed in 2015 between Alberta and the NWT. One of the goals of the agreement is to maintain the ecological integrity of the Slave River. Biological monitoring is needed in addition to water quality monitoring. Monitoring of Slave River fish under the Transboundary Agreement began in 2019 with Slave River and the Delta Partnership, SRDP, University of Calgary, University of Saskatchewan, and Wilfrid Laurier University. So large-bodied fish, lake whitefish, pickerel, burbot, jackfish were monitored in fall of 2019 and 2021 and winter of 21-22. Small-bodied fish, so lake perch, emerald, shiner, spot-tail shiner, were monitored in 2019 and 2022. Fish were sampled for length, weight, organs, weights, liver, gonad, spleen, age, and contaminants, PAHs, and metals. We also did a NASA project. So this project was doing satellite imagery of the Slave River Delta on water levels, land cover and vegetation, flooding and flood risk, flooding/landslides, water quality, and algae. In 2022 to 2024, they're looking at dissolved organic carbon in the Slave River and water quality probe deployment. Training and certificates. For all our programs under the Aquatics Coordinator, training is provided through each one. So at the start of the program, we have hands-on training such as the transboundary fish monitoring, the community-based monitoring, the air sampling, and Dr. Genois program. For the last 3 years, we have found funding to put on wildlife monitoring training, which is PAL, wilderness first aid, predator defense, and shotgun handling, land navigation, boat safety, Garmin inReach operations. These are great opportunities for our people to get training to work in the programs we put on or through the lands department for jobs as well, as these certificates help with applying for our on the water/land programs for the DKFN. And that's it for me.
Awesome.
So that's what we all do around. That's all our programs that we are doing so far and have done around here.
Awesome. Thank you so much, Kathleen. And yeah, sorry, I did miss at the beginning. I don't have all my notes pulled up. They're all over the place on my tiny screens. But as Kathleen mentioned, she's a third-generation monitor in her community, and as you might have been able to deduce from her presentation, she has insanely wide breadth of skills and knowledge, and it's very exciting for us to have here today. So my little outro/intro for you, Kathleen. Thank you. Next up, we're going to move on to Jeffrey Cederwall from the GNWT. Jeffrey is an aquatic quality scientist working on the Great Slave with the government of the Northwest Territories and the Department of Environment and Climate Change, and he is a Métis citizen from the traditional territory of Treaty 3 in northwestern Ontario, and now lives in Yellowknife, Northwest Territories within Treaty Draggese Territory. He has a wide range of research, freshwater research and monitoring programs, including diluted bitumen, oil spills, contaminants, including mercury and pesticides, fishery assessments, harmful algae blooms, and surface water monitoring for citizen and government science. And Jeffrey, I'll let you take it away from there.
Yeah, appreciate introduction and opportunity to speak to everyone here. So I want to start off by giving everyone just a quick introduction about the Great Slave Lake Sub-basin and the catchment area we're talking about. So this is a very large area transboundary, not just Northwest Territories, but includes inflow waters primarily from Alberta as well as BC, Saskatchewan. Our primary inflow to Great Slave is the Slave River, accounting for almost 80% of our flow, as well as some smaller rivers, including the Hay, Tulsin, Lockhart, and 12 other lower rivers. This all flows out through Mackenzie. However, we are not simply a river system. This is a large lake and needs to be studied as such. Great Slave Lake is the second largest lake in the Northwest Territories, followed behind just Great Bear, 10th largest in the world. And this is the deepest lake in North America, particularly in the East Arm section of Christie Bay. The lake is a diverse area where the main basin and East Arm are quite distinct lake types, where the more ultra-oligotrophic or low nutrient side of the East Arm is very different from the main basin of the open area that you'd see this section over here. This is an important fishery for both our commercial fishery, subsistence and recreational. It is the largest commercial fishery in Western Canada inland. Primarily this is lake whitefish and lake trout. And for the territory itself, it's over 60% of our residents in Northwest Territories live along or near the shores of Great Slave Lake. And I want to give a background as well onto what the brief history of the research has been on Great Slave Lake. This started in the mid-40s with studies led by Rawson, who did a whole lake assessment of this, including on the plankton and fish community, which helped set up this commercial fishery. And then during the 80s and 90s, there was some more study put into primarily focusing on contaminants. These contaminants were involved both within Yellowknife Bay from mining, as well as those transported to our region by both river and air. And more recently, Department of Fisheries and Oceans has restarted fisheries assessment looking at the fish movement, fish abundance, as well as the fish food. In 2012, the GNWT established the community-based monitoring program led by communities, which include sites on Great Slave Lake and the tributaries. And more recently, we have done a specific additional program on Great Slave Lake to address additional gaps within this and try to bring together everyone on the monitoring program. Let's go here. In comparison to other large lakes in the Northern Hemisphere, both Great Slave and Great Bear remain understudied scientifically. In comparison to our large lakes, Huron, Superior, Ontario, Erie, and Michigan are substantially more studied, being more southern and more accessible to study. And this is also in comparison to Lake Baikal, the deepest lake in Russia as well. So this is just a quick pull up from what are the research articles related to aquatic science and keyword searches for this as a comparison tool to show you the differences. So to try to address this, we want to work on a collaborative approach to make sure that we're understanding and doing what is needed and by communities. So started by trying to work from the onset with our partners, and this is involving primarily early and ongoing engagement. Some ways this has been done right now has been community engagement workshops, direct conversations with the communities, some ongoing community surveys, and collaborative sampling to get out together to see what's happening in the basin now and what kind of sampling we can do together in the future. The way we are developing this program, primarily as you develop any program, are 3 phases of scoping a program, defining a program, and implementing the program. To get this work undergoing now, different sections of this are undergoing at the same time. This has started with again, background research, engagement with communities to try set our priorities and trying to have those conversations about what should we monitor, where to monitor, and then building our baseline data and partnerships to grow this going forward. And in the future, we want to implement a more widespread lake-wide program that involves ongoing engagement with communities to ensure that we have the long-term sites necessary to monitor changes. Through our direct conversations with communities last year, this was led first through 3 virtual workshops with communities located on the shores of Great Slave Lake, as well as a 2-day in-person workshop that have been produced into summary reports to help guide the program. We heard a number of things from these conversations, which can help guide us developing 6 key pillars for program development. And first and foremost, this has to involve respect. And this is respect for a variety of knowledge systems and respect across relationships. And any successful program like this needs to involve youth. Both knowledge systems should be equally valued. This is both science and traditional knowledge. There are ways to best communicate with communities and with groups going forward, and we need to address the concerns and challenges within such a program to bring such diverse communities together, including capacity challenges that we're seeing. And I wanted to highlight that we can use both traditional knowledge and science together to get a more fulsome understanding of the aquatic food web here. And some key things that we've heard from these conversations is just the benefit of why we're doing this together and bringing both sides on this, and both the science and traditional knowledge, and the reasons that people here care about the water and want to ensure this is protected going forward. And we are seeing drastic changes in this sub-basin, primarily changing from the ice being different, which is having a number of downstream changes on the ecosystem now. But besides these direct conversations we've been having with people, we wanted to get another way to ensure that voices are heard and embedded within the program. And that's an ongoing community survey, which has been going. And this is a survey developed in partnership with Fisheries and Oceans, as well as Arctic Research Foundation, and to understand some more of the changes going on from key land and water users across Great Slave Lake. So this is asking, what have you noticed in this? What are your concerns in this? How do you best think you could be engaged on this? And what kind of priorities should a program incorporate on this? And hopefully these results can help refine and narrow the scope of a plan to initiate on the key priorities first and ensure that we can communicate back in the appropriate manner. So this survey has been completed by over 80 participants so far from a number of communities located along the shores of Great Slave Lake. And I want to thank all these participants to date so far and the local community coordinators that have participated in this. It takes over an hour, hour and a half to complete this. And without this information, we wouldn't be able to understand more in this going forward. We do have some preliminary results on this to share at the moment. One of the first questions is, what have you noticed in the water quality in Great Slave Lake? And we're hearing that there are generally is a more increased cloudiness in the water, more foam and bubbles, and more visible algae growth. And algae here is not noted as a periphyton, which is a attached algae, or a phytoplankton, which is a floating algae. These can incorporate both types here. And what are the largest human-caused concerns we're hearing? And for this, there's no single one concern that is the concern among communities, but these are widespread, which maintain to be mineral exploration, some forestry harvesting, climate change, as well as some legacy projects across the basin and downstream from oil and gas extraction and the oil sands development in the Athabasca region. When looking at when we're seeing the biggest change in Great Slave Lake, we're seeing that within the last 10 years, over 76% of participants are noticing that they are seeing the greatest change, not necessarily saying this is the only period of change, but the greatest period, with most of this occurring within the last 5 years. So what are we doing now for monitoring in the lake? So the GNWT with our partners have added additional new long-term monitoring sites, and these sites complement the existing community-based monitoring program that has been going on. So these are primarily located further from communities to fill in data gaps along Great Slave Lake in some sections of deeper basins and some important nearshore areas. We're monitoring with a combination of grab samples and continuous sensor deployment. And this is trying to sample the water column from the top to bottom. Over the course of the year, this is year-round monitoring with some samples taken in March and the sensor deployment year-round, collecting over 100 parameters such as temperature, dissolved oxygen, turbidity, nutrients, the major ions, metals, and chlorophyll A. Another new initiative this year are our 3 mooring locations, which are areas where basically it's a line of sensors anchored from top to bottom to understand the water column, and I'll talk more about that in a second. So our sample sites that we currently have located on Great Slave Lake now are included here. The yellow are the existing community-based monitoring sites supported by the GNWT, but led by communities. And we've added additional blue stations across sections of the North Arm and into the East Arm. And the 3 mooring locations being in deeper areas of Yellowknife Bay, the Hearn Channel in the middle here, and the upper area in Isti Bay there. So our program is designed to be long-term monitoring sites that continue to monitor going forward, but needs to be responsive to community concerns. Right at the moment, we are looking at the lowest water on record for Great Slave Lake, but not that long ago, we were at the highest water, and our turbidity was the highest ever in 2020. This led to a large number of community concerns regarding what is the potential impact on this on fish, what is the water quality, is this safe? So in 2020, 5 additional monitoring stations were added along the North Arm and into the edge of the main basin. These stations have been maintained going forward to today, and look forward to continuing them in the future. And I will highlight this was due to heavy rains that we had in the upper catchment area that increased our water flow through the Slave River, leading to the increased turbidity. Another newer concern we're seeing are increased reports of cyanobacteria blooms within the North Arm. This past year in July, we had a report of cyanobacteria within Yellowknife Bay area, in between Yellowknife and Deda. And we've been seeing this for the last probably 15 years on increased intensity and frequency through observations. We confirmed this is a species of cyanobacteria, which has been seen before in the basin, but never this thick in color. And we have additional research planned in partnership with Environment Canada, as well as with our Indigenous partners, to understand more of why this is incurring increased frequency in the basin and where to predict this. Another new initiative we're looking at that occurred this year are our Great Slave Lake moorings. And these moorings, again, are the deep water moorings occurring in about 100 meters of water, 300 meters of water, and 250 meters of water. And a mooring is simply a way to attach sensors top to bottom in the water column. And for our area, being ice-covered variety of the year, we are setting these 6 meters below the surface so that they will not be caught by ice. Can be deployed year-round. So a large number of buoys are added to keep system afloat, followed by a grappling hook for retrieval. And we have preset depths where we can attach sensors to a rope and weighted down by a heavy anchor of 200 pounds to keep this vertical. And should the grappling hook fail, we have an acoustic release system to ensure that we can receive back the sensors on a yearly basis to retrieve the data. Currently, for the pilot year, we have added just temperature and dissolved oxygen sensors, but have plans to add additional sensors to collect both water going forward and options for additional high-frequency sensors within the top layer of the water as well. So this was, our first one was deployed in April this year in Yellowknife Bay in partnership with the North Slave Métis Alliance. And I kind of want to walk you through of how this was done, as it's our first through the ice deployment. So we first got there and just verified the site depth that we had seen in the summertime to get the exact water depth that we're looking for. Once confirming the depth was the appropriate for our pre-measured rope, we then proceeded to drill a number of holes wide enough to fit our mooring buoy kit and anchors through it, chipping this out to smooth the edges on the side, scooping out the ice to not have too much slush so we can see clearly into the water. And then to deploy this in a very controlled manner, we actually attached this to a snowmobile. So the top, we deployed it in reverse manner with the anchor going first, buoys last, and using the snowmobile to kind of walk it forward, allowing us to attach sensors at our preset points and ensuring the sensors do not rub before going into the water. And then ultimately, as a group, deploying this, detaching from the snowmobile through the ice where it sits now today. Two other moorings were deployed, the second one being in Christie Bay in Deninu. This was in partnership with Parks Canada. And this one, the videos are not working today, but was at about 250 meters in depth, and the first time that we've done intensive sampling within the National Park Reserve. As well, given these large depths, things don't always go to plan. So in October, we redeployed our Hearn Channel mooring, which was actually set initially a touch shallow. So we went back to seeing that the buoys were on the surface, went back and pulled in this mooring prematurely so we could shrink the depth down to get our appropriate 6 meters below the ice, so that we could set it comparable to the other sensors. To summarize back on this, the data for these will be available starting next year, as they require a manual retrieval and reset each summer. And we do have options to add additional sensors for these based on what community priorities are, and options to maybe add additional locations based on both ecological areas that are important, such as deeper offshore areas, as well as near shore that important areas that are both cultural importance and ecological importance. But this program will be ongoing long-term in the future. And going forward, we will continue to actively monitor these long-term sites on Great Slave. The engagement with communities will be ongoing as well. Our community survey will hopefully be released to the public and communities first in the spring of 2024. We do want to continue to build our partnerships and collaboration to expand our program. And on our end, there is a plan to do a comprehensive assessment of what is the existing water quality information we have, what are the gaps, what are some emerging vulnerabilities for Great Slave Lake, again, to help better set our priorities. Through this, we're going to grow capacity on our program and ultimately develop and implement a full research and monitoring plan for Great Slave Lake. And this is focused on the water quality side. And again, with that, I want to thank everyone. And if there are questions at the panel, I'd be happy to ask them, answer them then. Or if you have any other questions, I'd be happy to take them by email. You can get in touch with me anytime. Thank you.
Awesome. Thank you so much, Jeffrey. Awesome. We have some emojis coming through. And yeah, folks, if you have questions like kind of top of mind, make sure to throw them in that Q&A. It also helps me plan out the session a little bit more. Robert, the webinar is being recorded, and you will be able to find it on our YouTube channel, which I'll post the link in after our last presentation, and I'll be sending out an email afterward. Next up, I am bringing up our final presentation, which is from Dr. Marlene Evans with Environment and Climate Change Canada as a research scientist in the Water Science and Technology Directorate, and she is based in Saskatoon. She's been conducting research and monitoring investigations of mercury and persistent organic compounds in the Great Slave Lake ecosystem for almost 30 years. And today in her presentation, she's going to discuss some highlights of these investigations, including community partners, and address national and international needs. And she's going to conclude with a mention of how climate change is impacting certain aspects of the lake ecosystem. But I will let Marlene take it away from here. Thank you. Oh, Marlene, I cannot hear you right now, but I can unmute you. There, you're unmuted. Oh, Marlene, I am having troubles with your mic. I can't hear you right now, Marlene. Enter request to unmute. Oh, Can you
Can
talk
you
now?
hear me now?
Yeah. Awesome.
Okay. Sorry for the rough start.
Your
So-
presentation right now isn't in full screen either if you want to put it there, but this is also good, I think.
How do I do that in full screen?
You know what? I think this works great if you want to put it there.
Is that full screen?
No, I do know someone said pressing F5 earlier today. We'll do that at the top of the keyboard.
If you can tell me how to do it, I'll do it. Otherwise, I'll just keep it as it is.
Yeah, I think F5 at the top of the keyboard might work, but if that doesn't work, then no. Yeah, just keep it how it is. I think this works great. I can see.
Okay, sorry guys. Let me just try one thing. No, I'm not going to play around with it. So thanks very much for this opportunity to talk about our studies on Great Slave Lake. As Carter mentioned, we've been doing studies for over 30 years, so we have a fair bit of data. So when I think of Great Slave Lake, I look at it from three perspectives. The first is Great Slave Lake is one of a series of large lakes spanning the edge of the Precambrian Shield to the west, to the east, and then Paleozoic deposits to the west. In the south, we've got the, In the south, we've got the Laurentian Great Lakes, and then as we go north, we've got Lake Winnipeg, Lake Athabasca, Great Slave Lake, and Great Bear Lake. Great Slave Lake is already mentioned as Canada's second largest and North America's fifth largest North American Great Lake, and it's the deepest lake. And it's interesting from the viewpoint that it's a relatively pristine lake. As Jeffrey had mentioned before, a lot of work's been done on the Laurentian Great Lakes shown down here in the south. And one reason is because there's a lot of people living there. Of course, there'll be studies where you've got people in universities based. But the other reason is there's been a huge amount of environmental impacts on the Laurentian Great Lakes. And these include eutrophication, overfishing, invasive species, and a large number of issues with contaminants. These really developed in the 60s and 70s, and there's a lot of remediation going on right now. But Great Slave Lake is quite different from the Laurentian Great Lakes from that perspective. It's isolated and it doesn't have the same development. The second perspective is when one considers Great Slave Lake is to recognize that it's part of the Mackenzie River Basin, as was already discussed. The major water flow into Great Slave Lake is through the Slave River. This water is not only massive in terms of water quantity, but it brings in a huge amount of suspended sediments, with these suspended sediments eroded from the river banks and the plains. So these suspended sediments and the chemicals that are associated with it have profound effects on numerous aspects of the quality of Great Slave Lake. And then because of the strong influence of the Slave River on Great Slave Lake, especially the west side, Great Slave Lake is vulnerable to activities occurring in the upper reaches of the Peace and Athabasca rivers, including pulp and paper mills, municipalities, and dams. So when one's concerned about protecting Great Slave Lake, one has to be aware of what's happening upstream in the watershed. And then the third perspective is Great Slave Lake is within the subarctic and Arctic region. And as we know, this area here is undergoing a lot of change. People right now are most familiar with climate change and warming and how that may be impacting different aspects of lake and river ecosystems. But before then, in the 1980s, early 90s, it started to be recognized that in the Arctic, there were a fair amount of organic contaminants in marine mammals, which were being transported into the north from long-range atmospheric transport. So for a lot of the research I do, it's part of monitoring associated with long-range transport of contaminants into systems such as Great Slave Lake. And our work is part of the Northern Contaminants Program, which is Canadian-based. And we contribute to the Arctic Monitoring Assessment Program, which is countries primarily in Europe. So if we look at Great Slave Lake and early contaminant concerns, as mentioned before, Great Slave Lake supports about 60% of the population in the Northwest Territories. So in terms of the Northwest Territories, it's probably the most important lake in terms of supporting different aspects of people's lives. Fish are a very important part of the lake, with the commercial fishery since the 1940s. Domestic and sports fishings were also important. And early in the development in Great Slave Lake, where the population started to be built up, mining played an important feature. And with mining development, just as in the Great Lakes, there was pollution. And some of the early studies on Great Lakes looked at some of these pollution aspects. Two general areas. One is giant mine. Mining activity with the gold mines in Yellowknife began in the 1930s. There were 3 different companies associated with this, but right now people tend to think mainly of giant mine. In the 1930s, there was a brief use of mercury, but that was discontinued. Effluent was released into Back Bay and into Yellowknife Bay. And for a brief period of time between 1949 and 51, large amounts of arsenic trioxide were emitted into the air. This caused all sorts of problems. And so, unlike a lot of other places, these emissions were very quickly dropped, so by the 1960s, they had decreased to 163 tons, and declines occurred even continue to this day. So in terms of contamination within Back Bay and Yellowknife Bay, it's generally located within 7 kilometers of the mine. It's associated with effluent release and then also with atmospheric emissions. If one looks at the sediment core record for arsenic, which is the compound of most concern, there were peak concentrations roughly corresponding to the time when the emissions were greatest. And then they've decreased since then. But research by John Chilliat and others are showing that arsenic is migrating up towards the surface of some of these sediments. And of course, soils in the area remain contaminated with arsenic. So arsenic contamination remains a continued issue of interest for the Yellowknife area. The other large mining development was a lead zinc mine at Pine Point, just near Fort Resolution. This operated between 1965 and 1998. More than 50 pits were dug. You can still see them today on Google Earth. There were 3 watering ditches draining into local creeks, and processed water was put in these tailings ponds, and there is some leakage from those ponds onto the landscape with significant zinc flushing. Only one environmental study was actually done on the terrestrial landscape, and they found that there were no aquatic invertebrates found near the tailings areas where the metal concentrations were high. But in terms of Pine Point, ore wasn't smelted on this site, so it was shipped elsewhere. So we're not seeing the same emission issues and other issues that we're seeing at Yellowknife, for example, with the gold mining there. I've been working at Flin Flon in Manitoba for a number of years, and where there's also a lead zinc mine. And it was only recently that the emissions from that particular mine were shut down. So because there was no smelting at the Pine Point mine, the environmental damage there is somewhat limited and primarily on the landscape. Long-range transport. They're interested in considering transport into the main body of Great Slave Lake is fairly recent. In 1979, a scientist called Allen, he hypothesized that mercury from effluent was moving out into Great Slave Lake, and might be seen in the spot here because he was observing an increase in arsenic concentrations in sediment cores. And Alina Mudrock did some other studies, and she thought possibly there might be some signal of emissions and effluent from Yellowknife into the main body, central part of Great Slave Lake. The other source of long-range contaminant transport is obviously through the Slave River, and the Northern River Basin study was conducted in 1991 and continued to 1996, and was designed to mainly investigate issues along the upper reaches of the Peace and Athabasca River related to pulp and paper mills. But other stresses were considered, including nutrient releases, and dioxins, furans, PCBs, and toxaphene were the main compounds that were looked at. A little bit of work was done on the Slave River Delta and on Great Slave Lake under this study, and I'll talk about that briefly. And then also in the 1980s, as I mentioned earlier, concern began to be raised about the fact that PCBs and DDT could be found in the Arctic in polar bears and seals at concentrations that were sufficiently high to be of concern to the well-being of the animals and also human consumption, with long-range atmospheric transport identified as the primary source. So that's when the Northern Contaminants Program was established to begin looking at contaminants and wildlife that are important in indigenous diets. And this program is quite broad. There are a number of sites throughout the subarctic and the Arctic, and fish, caribou, bird eggs, seals, beluga, walruses, and they are all being monitored. It's quite impressive. So let's see what my next slide is. So I just want to talk about some of the highlights of our studies. So, and again, quite a lot of this is old data. So over 1994 and 1996, we did a number of sediment studies. These dots here show the areas that we were in. In the summer, we had 3 trips with 3 trips in the area offshore of the Slave River, and then we got into the East Arm and sampled a bunch of sites there for sediments. We've also done sediment coring studies, always in the winter, primarily in the West Basin of Great Slave Lake. We tried to get cores in the East Arm, but we weren't successful because for a variety of reasons. One, when the bathymetry is not well known, and then I would say that'd be the primary reason right now, because we have got cores at near Lutzel Cay, but yeah, so I guess but they've only been looked at for mercury. So in the winter we've used skis and twin otters on skis and helicopters. The compounds that we've looked at over the years, we've had a big focus on mercury. We've looked at other metals, lesser work on PAHs, which are associated with the oil, and then legacy persistent organic contaminants. And we've also looked at diatom remains. The other thing is, sorry, just to go back. The other thing is we've got access to other data sets. We've got access to the oil sands monitoring data that was conducted under the RAMP program. There were the Mackenzie gas pipeline studies where there's data there that we've accessed. And then under our own Mackenzie gas pipeline project, we've collected data up in the Mackenzie Delta and on some lakes. So we're able to pull all those data together. So I just wanted to show you a couple slides. If we look at arsenic concentrations in surface sediments in Great Slave Lake, and this is a number of collections, we can see that arsenic oh gosh, this mouse is really sensitive. We can see that arsenic concentrations are generally between 5.9 and 16.9 milligrams per gram. About 74% of the samples exceed what's called a sediment quality, interim sediment quality background concentration. And this is just due to the fact that arsenic concentrations, background concentrations of arsenic tend to be high in the Mackenzie River Basin at a number of locations. The highest concentrations tend to occur offshore of the Slave River, where we've got a lot of sediment coming in, and it finds sediments and it settles onto the lake floor. We see lower concentrations in the East Arm, where not as much of the Slave River sediment gets into the that area. I sure there's another way to advance this. Let me just wait a sec here. Yeah, sorry, I'll use my mouse. So if we pull together all our data and look at Great Slave Lake in terms of the arsenic concentrations in the Mackenzie River Delta, which is in the upper left, we're saying, staying the similar pattern that with concentrations in the similar ranges. If we look along the Mackenzie River on the lower right panel, concentrations are low in the tributaries in a good portion of the rivers, but once you get generally within the broader Norman Wells area, the concentrations are just a little bit higher, and that's probably a feature of local sources, again, with groundwater and then just the geology of the area. And then for the lower right, I'm just showing data for Western Lake Athabasca and the oil sands. The areas where there's very low concentrations in the oil sands tend to be rocky, sandy tributaries. If we look at polyaromatic hydrocarbons, these are compounds that are associated with oil activity, and that's obviously important when one's considering oil sands development. And we see a same basic pattern where we've got higher concentrations offshore of the Slave Delta. And then as you go into the East Arm, concentrations drop off just because there's less sediment delivered into the East Arm from the Mackenzie River, the Slave Piece and Athabasca rivers. If we look at the Mackenzie Delta and some of the upland lakes, we see similar concentrations. There are a lot of natural oil sources in this area here, as there are along Norman Wells. So in the broader Norman Wells area, there's places there where concentrations are higher than upstream. And then the lower panel shows Western Lake Athabasca and the oil sands. So basically, we're seeing concentrations which in large measure reflect natural sources and are not at levels that would be of concern. And we're not, the patterns that we're seeing can be explained in terms of sediment grain size, organic carbon, and then proximity to oil seeps or bitumen deposits. We look at persistent organochlorines. We've looked at a number of compounds, or we did, PCBs, DDT, and concentrations tend to be, they're definitely on what would be considered the low side. It's very hard to detect these compounds in the sediments. And we're seeing a general pattern where concentrations tend to be highest off of the Slave Delta. And that's because we've got large amounts of sediment coming into the lake, and compounds coming in from the air are, brought down rapidly to the sediments. Whereas in the East Arm, we think we're seeing lower concentrations because the compounds that are getting into the water in the East Arm through atmospheric transport, they probably stay in the water column longer just because there isn't a lot of sediment in the water to pull these compounds down to the sediments. We've looked at time trends in, we can look at time trends in some compounds. Probably for metals, the time trend that is most interesting is arsenic, because there's been some speculation that arsenic from the gold mines at Yellowknife might make it into Great Slave Lake. But this core here, this is two cores, one offshore of Hay River and one offshore of Pine Point. And the two cores do show peaks in arsenic, very strong peaks in arsenic, actually. But the thing is, the timing of these peaks differ. One's at from 74 to 78, and the second one is a bit earlier, about 1960 to 1965. And these just don't correspond to the peaks when the emissions occurred from, the high emissions occurred from the giant mine. They seem to be more associated with a compound called manganese, which, for very complicated reasons that I don't fully understand, can sometimes occur in peak concentrations, either at some depth in sediments or near the sediment surface. And arsenic seems to be associated with that. John Chiliat's been doing some good work in the Yellowknife area, and he's seen the very same thing. But in his instance, the peaks occurring at the sediments. So we just don't feel that in terms of arsenic from giant mine having any detectable signal in the main body of Great Slave Lake. We're just not seeing any evidence for that. And it seems, yeah, so I'll just leave it at that. In terms of pHs, these are the compounds associated with oil. We're not seeing any time trends in pH concentrations in the core that we've looked at, which is closest to the Slave River Delta. So even though there's a lot of development occurring in the oil sands area, we're not seeing any evidence that it's having any impact on time trends in these compounds in Great Slave Lake sediments. In terms of persistent organic compounds, these are things like PCBs and DDT. We're not, these concentrations occur at these compounds occur at very low concentrations in the sediment core that we have looked at. And not really a sharp increase in concentration since the 1950s when these compounds started to be used. So I think that also is encouraging. We're not sure why we're not seeing a peak. It's only one core, but I think that part of the issue is we've got a lot of sediments coming into the lake with the Slave River, and it's a lot of eroded sediments. So it's probably masking a signal that we might see, and we might have more of a chance of seeing time trends if we were to get a core somewhere in the East Arm. Our main program is based on contaminant trend monitoring in fish. That's how our research started up in Great Slave Lake. We were able to get Great Slave Lake included as a site in the Northern Contaminant Program. We were able to argue that because of the importance of the lake to people in the territories and fish in particular. So we established two sites, and originally one at Litzel Cay in the East Arm, because the main source is atmospheric, and that is certainly what NCP is most concerned with. But we were also able to get a second site established in the West Basin, and initially we picked Fort Resolution because of our interest in the Slave River influences. And initially we hypothesized that we would see more contaminants in the fish near the Slave River than we would in the East Arm, because the West Basin gets atmospheric sources plus the river sources. Early on in the early years of the study, we got fish, and then we were also doing some food web sampling. We did water chemistry, but we were only able to do that for a couple of years. So in 1998, when the program was formally established, we're just looking at fish. We don't collect our own fish. We rely on the communities to get fish for us. And the study design is based on lake trout being sampled annually at Litzel Cay. We moved our sampling site for lake trout to Hay River because of the commercial fisheries. Lake trout aren't too common in the Fort Resolution area. We're monitoring burbot at Fort Resolution. Burbot is no longer, burbot was part of the program at Lusoke until about 2010, and then it was dropped, but we're still getting burbot, mainly for mercury analyses. We do get pike at Fort Res, mainly for mercury analyses, and we briefly looked at the Slave, pike in the Slave River. I'm sorry, burbot in the Slave River. In terms of our findings, this is on a log scale, so the concentration for every number goes up from 0.1 to 1 to 10 to 100 to 1,000. We're seeing, well, I'm sorry, I should go back. Watch the time here. So for our fish, we'd get length weight. We measure body weight, liver weight, and gonad weight. The fish are aged. The sex is determined. We measure stable isotopes in the fish to assess their feeding characteristics. The legacy pops that we're analyzing are PCBs, DDT, and HCH, among others. They're done in lake trout fillet and burbot liver. Mercury was added in 1998, and a few years later, a broader metal suite. And then as new compounds are being identified of concern, they're incorporated into our analyses. So we were looking at flame retardants and surfactants. And this year we're looking at new classes of compounds. And then other researchers are looking at microplastics in the digestive tract of our fish. And there's a microplastic study in water being conducted in the Yellowknife area. And as Kathleen mentioned, as part of our program, there's the community-run air monitoring station at Fort Resolution. Oh, geez. So in terms of our finding, we're seeing that concentrations generally increase from compounds like hexachlorohexane up to toxaphene. We're seeing higher concentrations in burbot liver, gets up to 900 micrograms per gram. Lower concentrations in lake trout fillet, and that's in large measure due to the fact that burbot liver are really fatty, lake trout fillet are not. And then our lowest concentrations are in northern pike. They're the leanest fish. Contrary to expectations, for burbot and lake trout, we tended to see, especially for the PCBs and toxaphene, we tended to see higher concentrations in the east arm fish than in the west basin fish. We're not quite sure why that's occurring, but we think maybe because of the clear waters, the PCBs and toxaphene coming in from the air, they don't have a lot of particulates that they can bind onto, so they're taken up more quickly by the fish. Whereas in the West Basin, they might become more quickly associated with particulates and not as easily taken up by not only the fish, but the lower components of the food web, the plankton, and then the small fish that the lake trout and the burbot end up eating. Concentrations in these fish are considerably lower than in the Laurentian Great Lakes, so they're considered to be low concentrations. And then as I said already, the lowest concentrations of organochlorines are in the pikes. So that's one reason why that monitoring discontinued. We were just filling in some knowledge gaps. In terms of trends, overall, we're seeing these compounds decreasing in concentrations in fish, pretty much at the same rate in the East Arm and the West Basin. The decrease has leveled off in recent years, and that's being seen in the Laurentian Great Lakes. That's probably due to a variety of factors, internal cycling and then food web dynamics. And our data go into reports that are large reports that are reproduced every few years by Canada and then by the AMAP program. I think I'm going to speed up here. So we're looking at trends in mercury also. We're seeing mercury concentrations in our fish are very low. They're well below, on average, they're below the 0.5 parts per million guideline. However, we are seeing a general trend of increase in concentrations in fish, although we're not seeing it in the sediment cores. The sediment core concentrations are pretty steady. We are seeing an increase in algal carbon, which appears to be related to warm temperatures. So we think what's happening for mercury increasing in fish, it's related to warming trends with lakes becoming more productive and the mercury that's in the environment more readily getting into the fish. We've done a number of special studies, especially with Fort Resolution. In 1996, we did a really nice study with them looking at water sediments and fish offshore of the Pine Point mine. And that was my first experience really working closely with communities. And it was really good to sit down and talk about a study design and go out and do the study. And then to have an opportunity to come back and talk to the community about our findings. We did a smaller study looking at organochlorines and different metals and fish organs. Sorry, I'm not talking very well. In 2010, we did some preliminary community-based monitoring studies, mainly using a YSI and GPS and SecuDis throughout the bay at Lutzo Cay and at Fort Res. And I like to think this has helped form the foundations of some of the work that's been done since then. The other study we did was a domestic water intake study. And Kathleen's already alluded to this, but we worked on it most intensely between 2001 and 2013. We did two things, or the community people did the variety, most of it, but they transcribed data that's being collected on the intake side of the water treatment plant. So there's daily measurements of temperature and turbidity, which are pretty fantastic for looking at climate variability and impact on water temperature in a specific region of Great Slave Lake. And then turbidity also with resuspension and strength of the Slave River flow. We also periodically collected water for turbidity and various metals. The nice thing about intake sampling is you can get water year round. And it would be nice to see that program maybe continue. Clearly, intake sampling is specific to a location, but still, it is valuable for trend assessments. And it's being used at a number of sites on the Laurentian Great Lakes. The other study that Kathleen mentioned were NASA studies. There was NASA scientists looking for a project to do involving remote sensing, and we were able to develop something with Fort Resolution to look at changes in the Slave Delta over the last few decades using remote sensing. And then there's a researcher now who's working with Fort Resolution, Hay River, and a few other communities looking at dissolved organic carbon. So great opportunities are being developed through a variety of means to work with communities on a variety of studies. For some reason that's not moving. I'm just showing this slide here again for the air monitoring. We've done special studies with Lutzo Cay. Lutzo Cay for a long time has been concerned about skinny fish in Stark Lake, where there was a uranium mine, an exploratory uranium mine. We basically looked at a variety of lake trout with them, and we did see they did provide us with some skinny fish. They tended to be older fish and a bit higher in mercury. We think fish get skinny when they're either very sick or they're near the end of their life, and they will have higher mercury in them. And the only other thing I wanted to mention is when I first started coming up to Lutzo Cay and Hay River and Fort Res, nobody wanted to talk to me about my contaminant studies. They kept asking me, what about this satellite, Cosmos 954 that crashed over Great Slave Lake? What did people find out about that? So we were actually able to, as working with Lutzo Cay, to do some more research around that. We found this really cool article called Operation Morning Light that really describes the explosion, then the search and rescue, or the search that people did to look for the debris. So if anybody's interested in this and hasn't heard about this report, I'll be glad to share it, because it's yeah, that was pretty scary for folk, people showing up in communities in yellow suits. So yeah, so conclusions, I'm just going to apart from primarily the Yellowknife area, we're not seeing high concentrations of contaminants in Great Slave Lake. There are some issues remaining with Yellowknife, concerns still with Pine Point Mine. And then, as Kathleen mentions, there's some concerns around the inner part of Resolution Bay, but in part with some local releases. We don't know a lot about the influence of the Slave River on Great Slave Lake, and a lot more needs to be known about how it brings in sediments and nutrients and contaminants into the lake, how these spread out into the lake, how they get down to the lake floor, are they resuspended, and how they go through food webs. I think I'm just going to end here because I see I've been yakking for quite a long time. Yeah, so I'll just end here with communities are becoming increasingly involved in studies related to Great Slave Lake. They're developing their own community-based water quality monitoring programs in addition to participating in programs. I do feel that sometimes these studies can be a bit overwhelming to communities with so many researchers coming into the area. And I think there is a need to, one way or another, to have more full-time trained staff available within these communities to help oversee these programs. There's obviously an interest in more training and certification. A number of locations have field camps where they bring out elders and youth and researchers to talk around issues. And maybe it's timely now to have more of these camps for things specifically related to Great Slave Lake. And there's a need to bring in concerns or issues with Great Slave Lake in college and university courses, and maybe even having field camps and classes taught up around Great Slave Lake. So I'll just end there.